President-elect designate. So he is, I'm sure, extremely busy these days. And appreciate him taking time to come talk to us. You probably have also seen him through many of his other volunteer activities that are literally too numerous to list here today. But been very active in imaging research, as I think many of you know, mammography and CT as well, and of course the MedFIS 3.0 initiative. So a man of many, many talents, and look forward to his talk, which is on the role of virtual clinical trials in image-based research and practice. So Dr. Samei, all yours.
Hello, everybody. Can you hear me okay?
Yes.
Great. Thank you. Again, I apologize. I was just 1 minute late to my talk because I had a good excuse. I was actually chairing the MP 3.0 committee. And just like I told them, guys, I have to go. But it was just, anyway, I apologize for that. Thank you for, your patience. I'm really delighted to be with you. I'm really sorry that I'm not with you in person. Maybe that would be remedied next year. We'll see how the pandemic goes and so on. So I am here with you not in my capacity as the FAAPM President-elect designate. I think that's my official title now. But rather just as a medical physicist, and I was excited to, but as one of the latest things I've been doing in my research lab, which hopefully would have some relevance across board, whether you are a therapy physicist, imaging physicist, clinician, researcher, government person, or whatever. Let me share my screen. Make sure you can see my screen okay. Is this good?
Yes, we can see it.
Excellent. Ampere, one other quick question. How much time do I have for our whole time together?
Let me see. I think we have until, so we started 10 minutes late, so we have until 2:25.
Excellent. And that includes question and answers. Okay.
Yeah.
I have about 40 minutes worth of material, so we would have about maybe 10 minutes at the end. I would encourage you to sort of tighten your belts. It's a tour de force of this topic. I go through my slides rather quickly. I am a fast speaker, and feel free, of course, to jot down questions that you want to ask me at the end. So the topic is the role of virtual clinical trials in image-based research and practice. And first of all, I would like to acknowledge members of my lab and my colleagues here at Duke who has participated in many of the material that I will be showing you today. And also up front, I would like to acknowledge what a week it has been. Earlier this week, we celebrated the birthday of Marie Curie, International Day of Medical Physics, discovery of X-rays, International Day of Radiology, and then now the National Radiologic Technology Week. So it has been a heavy sort of radiology and medical physics week, and I'm honored to be sort of part of your activity this week in this very summit type space that we have together. What is virtual clinical trials? You might ask. I hope I will answer that question by the end of this talk. I just want to point out that the Journal of Medical Imaging last year had a special issue on this topic, which I encourage you to look at it. There was an editorial that I wrote, and then there is also a review article in that space. Here is the outline of my talk. I have a rather hefty intro, talk about what is a virtual trial, and why do we need it, and what am I doing with it here at Duke University. Spend most of my time talking about the science of virtual trial, and at the end we talk about some of the applications and future prospects. I'm going to walk you through this outline. So let's talk about the intro. So there has been a rapid rise in the number, in the complexity, and the variability of technologies, both in imaging and on the therapy side. And they really outpace our ability to optimize their design and clinical use. I'm just demonstrating here the number of technologies that has been introduced in CT imaging alone. You see, every few months, there is a new thing that comes to us. How are we using them? How can we be certain that we are using them clinically appropriately? Well, we often don't. FDA has approved the device, we are putting it in the clinic, and we sort of work it out. And we hope that we don't find major surprises in our first few patients. That's not really good enough. We need to make sure that these devices are well positioned and well optimized for clinical use. Well, how can we assure that? We can do experiments, what we call clinical trials. The problem is that clinical trials take time. We are out of time, stepwise out of time, as well as cost to actually do these. There is also some certain level of patient risk and uncertainty of truth in the process. Just as an example, DMIS trial was a trial that conducted a few years back in the US, looking at the effectiveness of digital mammography. That took $26 million to accrue 50,000 patients. And by the time they were done in 4 years, all the technology that they tested were outdated. So you cannot do a trial at the pace that technology comes to us. So what do? We do what we always love to do. We use phantoms. But the problem is that phantoms provide some reflection of the reality, but they are often too simplistic, simple, and cannot be extrapolated easily to clinical utility. At the end of the day, we say the technology needs to be used clinically to be able to be certain that everything is right. So you actually can think about, there is a problem that we have. We would like to do experiments on actual humans on the right side of the spectrum, doing clinical trials with actual human patients. The results are highly relevant, but they have very questionable practicality. Alternatively, you can evaluate technologies on the left side of the spectrum using phantoms, specifications, and so on. Those measurements, those approaches are highly practical, but they have very questionable relevance based on what I just said. So virtual clinical trial and virtual imaging trial is an alternative, is a midpoint in the space. And we like to argue that is more toward the right side than the left side, that we argue that can be both relevant and practical. So what is the caveat of doing this imaging trials with computational patients? That's what virtual trials are, is that you need to do it with realism. That means you have anatomical realism, technological realism, and perceptual realism, how those images, if you do in the context of imaging, would be used. Otherwise, you would be simplifying the problem too much, and your simplified solutions would not be indicative of what would occur in live experiments. And you also need to do it comprehensively. If you want to do a trial, you need to do the whole spectrum, from the patient to the imaging system to the image analysis, the entire spectrum of the things. Otherwise, we only have partial answers. So what is the virtual imaging trial? Is conducting a clinical imaging trial virtually. So in the actual trial, you have real patients with real acquisitions or interventions, and real clinicians interpreting those results. In the virtual domain, you have virtual patients with virtual acquisition interventions and virtual interpretations. So you can say this is an opportunity for medicine to meet matrix. So based on that, we make an argument to the NIH to sponsor a center that was launched at Duke University called Center for Virtual Imaging Trials. The purpose of the center is to develop and provide a virtual platform to assess the clinical performance of medical imaging systems from design to use, all the way from design to use. We are joining 24 such resource centers nationwide. There are many of them out there. None of them really focus on virtual trial. We are the only one that does that. It's made of 5 cores, 3 technical, 1 training, and 1 administrative core. You can think about each core being like a little R01 of its own. And then we have 22 collaborating partners of projects. And they come from, and there are CPs, they call collaborative projects, that interact back and forth with us, or there are service projects that only they are recipients of the goods that we produce. And they come from academia, they come from industry, they come actually from the government itself, and they come from societies. And we have an internal advisory committee that orchestrates and oversees our process, and we have an external advisory committee. And here is the org chart of our administration, if you will. I am the director of the center. Dr. Seegers serves as the deputy director. We have a whole host of administrators and computational programmers and so on. And then we have each one of those core groups that I mentioned have its own lead. It's a multi-R1 project, if you will. And we have collaborative projects and service projects. And so if you really count the number of people that are involved in this, there are close to 100 people in this space. It's a growing community. And within that community, each person has a different role. So we have different individuals for each one of those cores that are orchestrating and doing different activities for different things. These are the members of our internal advisory committee. Some of those individuals might be known to you. For example, Dan Sullivan, who is now a professor at Duke, used to be at the NIH, led the NCI's imaging program for many years. And each one of them really a who's who in their own respective domain, ranging in clinical domain to cardiology to AI, clinical trials, and so on. And these are the members of the External Advisory Committee. Again, these are who's who individuals with very deep expertise in CT, in imaging science. Dr. Kyle Myers used to be head of the major laboratory at the FDA, is on our committee. Dr. Ron Summers from the NIH, a leading AI scientist, and so on. And here are our collaborative projects. You see they're all over the map in terms of their institutional affiliations, and they're all over the map in terms of the applications that they're using. So they're using our resources, for example, to figure out how we can robustly measure radiomics features, or how we can do a better optimization of treatment of cardiac lesions, or how can we orchestrate PET-CT imaging better? How can we optimize photon counting? How can we use xenon for detectability of brown fat, phase contrast imaging, and so on? And here are service projects, again, a whole range of topics from emphysema to motion compensation in radiation therapy, optimizing 4D CBCT, and so on. So there are a whole host of different applications. And as a part of this center, we're going to create a series of software packages that, for example, ideally, when we are done, we are still in the middle of this work, you can go to this platform and say, I would like to have 100 patients of this age, weight, race, sex, profile with this type of abnormalities in them. Please simulate them for me. And then you say, I would like to image them according to these particular protocols that have been specified. And then analyze the results based on the things that I'm interested to learn from these images, and tell me whether modality A or modality B is better, or way of doing protocol A versus protocol B. You can discern that stuff together. And just one quick, the last slide of intro. In the last 6 months, we have launched this new national center.
Could you try again?
Sorry, my Siri is talking to me. Launching the new center in 6 months. We have started monthly forums that you're welcome to join every second, I think it's a Friday of the month. I apologize for that. We have a cyberspace presence, cyberspace webpage, social media presence. First set of software packages are released now. We have a residency program. People can come and spend time with us. We have so far published 10 papers in the last 6 months, and we have 11 more under review. We have 20 abstracts accepted, plus another 22 under review. And we have given 7 invited talks, including this one. And then plan ahead is that we have a grand challenge. It's going to be launched through the AAPM actually in 2022 on truth-based CT reconstruction, and courses at the SPIE and AAPM. You can actually come to the AAPM and learn more about this. And here is our webpage. You're welcome to check it out, cvit.duke.edu, and there's a whole host of details there that you can find out. Just a quick feedback from people listening to me. Am I talking too fast, too slow, on the right pace?
Sounds great.
Okay, great. Okay, so I'm going to walk you through some of the science of this. So the first task of doing virtual trial is to actually have the right patients, right? So the first thing that we do is create the patients. We are modeling the anatomy and physiology of the patients. We are trying to model the entire organ system, and it's based on the earlier work that was done. Dr. Paul Seegers, my colleague here, the co-deputy director of the center, that we essentially have these XCAT models. They're dynamic models, so we have a breathing patient and heart beating and so on. So those are all converted, and we have not just only one, rather a host of these models. In fact, we have zillion of these. We have adult patients, we have pediatric patients, we have pregnant patients, and we have even ICRP reference XCAT phantoms modeled. How do we create these? We have a pipeline that we call iPhantom. Hopefully, we would not be sued by Apple in the process. The pipeline essentially takes images, actual CT images of the patients, and automatically, through machine learning, create a digital twin of each one of those patients. We are segmenting major organs, and you see how well we can do the segmentation. Of course, that by itself, I'm opening the hood, is not perfect. In fact, we are like one of the things we are doing at the center to figure out what's the right type of segmentation we should use to have least amount of error in segmenting various organs. And here's I'm showing you Dice coefficient of how well we can segment various organs using two different segmentation algorithms. The work is going to be presented at the SPIE next February. After we segment major organs, then we deform one of our existing XCAT phantom to match it. And then with that matching idea, we fill the other organs that we have not been able to segment. The problem with segmentation is that you cannot segment everything in the body. So we use one of the more complete models. For example, if you're not successful to segment pancreas or prostate, we morph one of the existing model and fit those features back in, and that would create the new XCAT phantom. So one of the major initiatives at our center here to make sure we have the right level of anatomical variability. If you want to do a trial, you don't want to have only one patient. You want to have patients that would represent a variety of the patients that we see clinically. So far, we have segmented 7,000 models, and we are looking at their age and gender and size and race to see they represent the variability that we see in the clinical platform. Here is just a sample of, for example, liver volume across white patients versus Black patients versus other ethnic groups. And the idea is that you have to create a mold, a population, basically, a virtual population that reflects the real population that is out there. Otherwise, you cannot do a trial and you cannot trust the results, right? One of the major initiatives that we have is that we would like to create this data for training AI algorithms. So as you know, AI algorithms are very data hungry, and they are at the mercy of the diversity of data that you use to train them. So if you don't have enough diversity in the data that you're using for training, then you are not really any good. You're like a phantom, a physical phantom type experiment. So in this process, we do not just the major organ, but also the tissues inside the organ. So here, for example, we can texturize the lungs, and you see we can create texture for the bone. We are adding additional features such as the lymphatic system features and so on. Here is how we are doing the respiratory model. We have a complete model of the lobes, the airways, veins and arteries, and the parenchyma of the lungs, lung parenchyma, as well as looking at essentially the ventilation maps within the lungs. Here, for example, you see how we have used 4D CT to look at the ventilation map and implant them inside these XCAT phantoms that are much more realistic in the middle one to the real ventilation map that we have in actual patients on the right side. We have a dynamic model of the perfusion. So essentially, we can see what happens if you inject contrast agent to the patient and see at what time the patient, the contrast arrives at various organs inside the body. Essentially is ordained by a differential equation. It's out of a series of differential equations from which the solution would be essentially the time enhancement of the contrast material inside various organs. So we can do contrast enhanced imaging. And then along the same line, we also go even one level deeper, is that can we sort of look what has happened inside each one of the organs. So we have actually mathematically creating vasculature inside various organs. So far, we have done it for the liver, we have done it for the brain, and we are extending it to kidney and other organs using this sort of network of vasculature that is inside each organ, so we can do that enhancement at the micro level, if you will. We have a growing model of tumors. This is just one example. And the tumors are heterogeneously formed. So you can, for example, place these tumors at various locations inside the body if you're interested. And you can define different levels of heterogeneity or different growth patterns that you're interested to investigate. So we can, for example, we can see how the lesion is moving, how his lesion is growing, whether your evaluation is reflective of the truth of the growth of the tumor or not. You might ask the question, how realistic are our tumors? This is a study from years back, actually. We actually did an observer study with actual expert radiologists, showed them real cases, real lesions, and simulated lesions. And basically, they could not differentiate between real and simulated. The ROC study that we did gave us a AUC, area under the curve, of 0.5. If you had done ROC studies, you always gravitate, you're trying to get the ROC closer to 1 as possible, because you're trying to differentiate. Here, we're trying to say, can we confuse the radiologist, what is real and what is fake? And we are able to confuse them because our simulated lesions are so realistic. We are modeling additional abnormal tissues. We have a model of the cardiac plaque, and I will talk to you in a second. We have a model of COVID-19 now. We have a model of COPD. We have, of course, models of various fractures inside the bones. So we can place them. We can create these abnormalities, if you will, inside our patients. We have a complete model of the heart. It's a dynamic model of the heart, and we can actually alter this dynamic. And we are trying to model various schemas. Not all of these are completed, by the way. I am showing you a prototype of the heart that we currently have, that we are trying to create a library of putting various heart inside various patients. Here are models of the plaques. We start with segmenting real plaques, and then we use GAN to generate synthetic plaques, if you will. And then we can place these plaques inside the vasculature of the heart. And we're also working on a model of the stent to see, can we essentially create this virtual patients of ours. Here is an example of how the phantoms look like. This looks like a CT exam. It is not the CT. This is the ground truth of the phantom. This is the real patient, virtually defined, of course. And you see how the vasculature inside the thorax are affected by both the respiratory motion as well as the heart motion. Now, of course, if you're a therapy physicist, I'm sure your ears are perked up, because that gives you a ground truth way of evaluating whether your motion compensation strategies are effective in a way that you actually know for sure. So that was virtual patient. After we get the virtual patient, we need to essentially model the imaging process. In the first phase of our grant, we are focusing primarily on CT. But in the future phases, hopefully we gravitate toward other modalities as well. Here now, in the same way that I would like to make my virtual patients as realistic as a real patient, I would like to also make my virtual scanning as realistic as a real scanning, at diverse scanner models. For that, we have created a new simulator. We call it DukeSim. It essentially uses ray tracing to create high resolution images, and then we add a Monte Carlo for accurate scatter and dose. You might think, as a physicist, why don't you do the whole thing for Monte Carlo? You can't believe how many photons are involved in creating a CT image. It's still amazing how far we have come in computational resources. We still cannot do a single simulation of a CT scan through Monte Carlo with all the GPUs out there. So we still need to do this ray tracing supplemented to Monte Carlo. We still use GPU acceleration for speed, and then we are trying to do all of these in a manufacturer-specific fashion. And it's a demonstration on the bottom On the left side is a ground truth of the real, digital, real as it takes in the virtual domain of the patient, with the tumor, for example, is magnified. On the right side is a state-of-the-art CT scan of that patient. Here you see the dilemma of imaging. We reflect the reality of the patient, but we don't do it perfectly. We do it approximately. Imaging is about the art of approximation, if you will. That beautiful speculated lesion out there is blurred, is deformed, is noised in the image that we have on the right. With the virtual domain, we know exactly how much is noised, how much is blurred, how much is deformed. And then we can figure out what level of deformation or denoise or noising or blurring is acceptable without compromising the overall value to the picture. So in order to do this modeling as realistic as possible, you have to open the hood and model every element that is involved in the formation of the CT image. So I need to have the geometry lined up exactly the way the manufacturer does, the bow tie filter lined up, the subsampling of the focal spot, subsampling of the detector, the spectrum, the way that detector deforms the signal that is received, how the images are reconstructed by the manufacturers to create a clinically relevant image. All of those are essentially needs to be modeled exactly as is done in the actual CT scan. And that is what we do. Essentially, we have right now, as of now, we have 8 such models are generated or in the process of being generated, including a generic scanner that we have defined, and including the latest photon counting CT system, the first clinical CT system that was approved by FDA at the end of September. If you just happen to have missed that announcement by the FDA, we happen to have that scanner actually installed at Duke University, and we actually have a complete model of that scanner already. Here, I'm showing you some examples of, for example, a simulation of a lung lesion using GE Revolution and another photon counting system called from Prismatic in Sweden. We are able to create those images. And here are alternative images from a Siemens Force and Siemens Neotone photon counting CT system. So we can essentially image the patient and do a comparison of how one imaging system or protocol compare to another. And if you're more medical physics phantom oriented, here is a phantom image, real image on the top, real images at 3 different MAS levels, and simulated images on the bottom. And you see the images look very realistic in terms of, if you do an observer experiment, you can't differentiate which one is real, which one is fake. Again, you're a physicist, so you need numbers. And here are the numbers. We actually did a validation, for example, of Siemens Force across 2 current modulations with our phantom, and we measured the noise magnitude as a function of size. And you see the blue is the simulated data, the red is the real data. Close approximation. This is how the MTF lines up across different settings of the 2 current modulation. Here's the noise power spectrum or the noise texture. And here is the HU values for different types of inserts, iodine, bone, water, air, and so on. And here are our latest results from Neotone modeling, that photon counting CT system that is now FDA approved. So the green, I showed you this table a little bit earlier. The green areas are the areas that we have already modeled in the lab. So we are able to do a complete simulation of the Siemens count or Siemens Force or Neotone photon counting. And we can do models of G750 and G Revolution and Prismatic and so on. And we are advancing that space. So I got the virtual patient done. I got the virtual imaging done. Can I read those images? Well, you could put those images in front of a clinician and say, what do you think? But if you're doing everything virtually, why don't we just do virtual reading as well? Well, you could. So the point of this virtual reading is to model the image characterization and interpretation process. So when I say reader is not a one reader, other virtual readers, there are actually 3 different ways of approaching a medical image, whether being virtual or real. you can use observer models, essentially modeling the radiologist task. So here showing you under the hood, here, for example, we have done various observer models. On the y-axis is the actual detection performance of radiologists. On the x-axis, there are different observer models. And you see there are different levels of correlation between the observer models and the real detection performance of radiologists. The good news is that they're correlated. The bad news is that there is noise in that space, right? So when I say I want to model the interpretive process, it's not perfect. That's why I label this slide science. There is science to be done here. Which one of these models are the one that we should trust more for the detection? The second way of reading an image is to apply mechanistic quantification of the imagery data, radiomics, that you are very familiar with, whether in the imaging domain or therapy domain, and what are the characteristics? Can I have an algorithm that reads my virtual images? And finally, probably the most significant elephant in the room is AI-based characterization. We are now increasingly relying on, or we're hoping to rely on, AI algorithm and deep learning machine algorithm to read the images for us. Well, essentially can be make sense of the images through machine learning. And we are trying to develop methods to be able to do so, considering the fact that this is also already still an emerging area of work, that more work needs to be done in that space. One area that also be focusing on this reading space is called image harmonization. So it turned out that especially AI algorithms are very sensitive to this, that there is whether the data comes from one vendor versus another vendor, from one protocol versus another protocol, the AI algorithms get confused in a way that they get more confused than the human observers. In fact, I would say the radiologists and clinicians have already developed methodologies in the brain to compensate for the diversity of appearance of images. Of course, diversity in the patient images is okay and appropriate. In fact, that's how we make our processes patient-specific. That diversity is good. The diversity because of the way we treated the patient, because the way we image the patient, that's a diversity we need to get rid of, right? So we can essentially have a methodology to harmonize the data. So here is, you can have a non-harmonized image on the left, which would look into a reference look that we are interested in, and then it harmonizes that image, post-process the image to the particular look that we are interested in. This is a task that actually, I think, overdue, forget about AI for a second, or virtual domain, is a task that is overdue across our imaging enterprise for real clinical imaging, because we have this diversity that we have to somehow manage to make sure our care are much more patient-specific and a little bit less technology-specific. So at the end, I just want to leave you with some examples of now having these tools at our disposal. What do we do with it? Again, we live in a loaded world in which there are so many things to look at and pay attention to. So I have to subsample that space for you. So hopefully the examples that I'm providing would be relevant to some of you. Number one is that can be estimate the organ dose across CT protocols. This is actually something that you cannot do in real patients anyway, because you cannot put probes inside the patients. So I can get, in this case, 3 patients, pediatric patients, and I can scan them with chest exam and abdominal exam and see how the dose will vary across the body habitus or across organs. Or you can see if I invoke the two-prong modulation or use a fixed MA, how the pattern of dose deposition will change. Of course, you recognize for all of these examples, there is a spiral pattern because we are modeling a spiral CT. That's why you see the dose distribution is not uniform, as you expect. The second example would be, can we do individualized dose estimation? Yes, we can. So you can get the scout or topogram of the patient and match it to one of the patients in our library. And if you do so, then that library model can become a model of the patient that you have, a digital twin of the patient, if you will. And then you can do imaging of the digital twin, and then you can get the organ doses lined up. Now, this technology actually has been around for a while, and we actually is already licensed. We did this a few years ago to GE and Immunologics, and you see how it performs across different protocols. You see we are able to keep track of the 2' modulation, which is critical for organ dose estimation because MA is not fixed as you do a CT scan, and you can see how you can essentially decipher the organ doses. The second question that we tried to answer with our virtual domain was the plastic appearance of iterative reconstruction. So iterative reconstruction in CT in particular has been very popular. Now is advancing into deep learning reconstruction. And it reduces the dose dramatically. You see the demonstration here, a filter back projection on the left and iterative reconstruction on the right. And you see that the images on the right is significantly less noisy. But radiologists have always complained there is something weird about this iterative reconstruction. They refer to them as plastic appearance. It's like, what is that? We can't really, our measurements do not quantify that. So, well, we set to actually quantify that. What we did is that I got my patient and I imaged it 50 times. So if I do it 50 times, I can look at the ensemble of noise across those 50 repeats. It's not noise in the area, is across the ensemble of images. And now if you do that, you see a different pattern emerges. You see the noise on the left side is rather uniform because it's basically quantum noise. But on the right, noise is not uniform. There is more noise at the edges than the uniform areas. So it's true that iterative reconstruction reduces noise, but only does so effectively in the uniform areas of the image, not at the edges. And that's precisely what that gives that plastic look to the iterative reconstruction images. In fact, I have quantified that in this slide over here. You can see how much actually noise increases, not actually decreases, increases at the edges. So background noise has gone down, but the edge noises goes up when we invoke the iterative reconstruction. We can only do this with virtual trial because no patient would allow you to image them 50 times. I guess you can do in cadavers, but that's much more complicated if you have ever done those kind of images before. Yet another example, we like radiomics, we measure quantities such as volume or heterogeneity or entropy or speculation of lesions. How realistic are those measurements? Well, we can create a lesion, such as the models that I'm showing you here, and then we can take one of them, for example, and image it across a zillion different ways of imaging that patient and see how that lesion presents itself, right? So we did this 200, and by the way, you can also image that patient zillion different times, what we call a coffee break simulation. Imagine the patient comes in, gets in the scan, go out and have a coffee and come back. Image them after the coffee break. Would you get the same measurement? So we can do that. So we did this essentially 250 across 250 different protocols multiple times. Here is a complicated plot. Each row is a different imaging protocol that uses different dose, different slice thickness, different kernel. And on the x-axis, you have different radiomics features, from radius to volume to elongation to flatness and so on. And now I am showing you the bias in your measurement, whether you got the volume right, whether you got the radius right. Now, can I speak to bias? Yes, I can, because I know the ground truth. You can never get this data in actual clinical cases because you never know the ground truth. Even if you open up the patient with surgery, you do, in the process of doing so, is like Heisenberg principle. You're going to deform the lesion, so you never know the ground truth. So this is bias, this is variability. Now, if you, for each one of those images, if each one of those protocols rather, you repeat the scan again, would you get the same measurement? Well, you see generally when the obvious, when the dose goes up and when the slice thickness goes up, the level of variability would go down. But you see, based on this, then you can figure out what protocol I ought to be using to make my imaging process more quantitative, not because I'm guessing so, because I'm knowing so, right? So we can essentially do that. So we did an experiment in which we allow all protocols activated and see how we can quantify minimum detectable change in a particular aspect, radiomic feature. And if I only use the protocols that are ordained by Quantitative Imaging Biomarker Alliance of the RSNA, you can see I can be more targeted in quantifying that attribute of the lesion. So I can be more quantitative in my practice of medicine. All of that is made possible through virtual imaging trials. And now we are extending that work, for example, in quantification of COPD with CT. We need to have an imaging way to quantify the phenotype of COPD. Here I'm showing you the ground truth on the left side, and across 3 different types of models of CT scanners, Siemens Flash, Siemens Force, and Siemens Neotone. I'm using a soft kernel to form the image, or I can use a sharp kernel to form the image. And I can see how my quantification of LAA95, which is one of the biomarkers of COPD, changes. So I can figure out how my instrument can be made more quantitative to best serve the patient. This is yet another example of our work that we are doing on long COVID. So the idea here is that we create virtual chest radiographs from simulated patients with COVID-19 progression implanted in them. Then we essentially generate images of diverse imaging conditions that you might see in the clinic out there, whether what part of the country you come from and what imaging system that you're using, you will have a slightly different version of the same image, if you will. And then we can invoke our harmonizer to essentially make image appearance consistent across different diversities of appearance. And then you can feed that into an AR algorithm that discern the progression of the disease across longitudinal data. And hopefully at some point we can essentially create a visualization of the data to say not only whether you got COVID or not you got COVID, you can get that with genetic tests. Rather, what is the state of the disease in you? And whether it's progressing, regressing, how are we doing moving forward? Last couple of slides, and I will wrap up. A lot of work has been done, but there are a lot yet to be done. What are the priorities to move forward? Well, we need to enhance the realism. We have come a long way from having a piece of plastic represent the patient. Our phantoms have done great work. We are getting better and better, but we need to have more patient and task variability, more intra-organ pharmacokinetics. We have done it for some organs, but not all organs. We need to add more texture and pathologies. We need to do a complete model on the physiological-based motions as we are doing, vendor-specific simulations, and modeling the interpretation processes. AI interpretation, as I mentioned, is already an emerging field. It's changing, so we need to change along with it. And then we don't claim that we would fully emulate the actual clinical reality. How close is close enough? We need to answer that question. And we are thinking about using task-based validation with actual trial. I do my trial, I do my virtual trial. If they match, we'll say, well, we're close enough. But that's not good enough, because at some point I have to extrapolate from the models that I have validated to the ones that I am not going to validate. Because if I'm going to validate every virtual domain, then why am I actually doing the virtual anyway? I'm going to do the clinical trial. So we have to figure out how much we can extrapolate and interpolate our predictions of the virtual trials, and coming maybe some sort of information theoretic modeling of the closeness. And also, in terms of how close is close enough, I should also acknowledge, even if you do 2 trials back to back, you don't get identical results anyway. So there is some degree of grace that we don't have to exactly replicate the reality of every single virtual trial. There is some variable in that space already built in. And then we have to apply this further to research and clinical needs. So let me leave you with 2 slides of take-home points. Number 1, healthcare reality necessitates practical alternatives to clinical trials. We just cannot do a trial for everything that comes to our clinic. So VIT is a solution to our limitation to conduct in vivo experiments. In fact, I would argue that's one of the major limitations in medicine. We cannot do experiments. That's why medicine is so slow to progress. The rapid rise is a solution to rapid rise in the complexity and variability of imaging technologies, which outpace our ability to optimize their design and use, answer the question of cost, uncertainty, time commitment of actual trials, and our need for relevant and practical means as precursor or replacement for clinical trials. And VIT is a platform for all of us to do new science in medicine, to design and condition imaging for best clinical use. However, effective virtual clinical trial requires realism, effectually enough to predict clinical performance. These words are perfectly chosen. Is not needs to be perfect. It needs to be effectually enough. Needs to be close enough, and needs to represent the diversity of tasks and variabilities of backgrounds to garner generalizability. I don't want to do a trial and you come at the end to tell me, you know, your patients are just too clean. They are not reflective of the reality of the clinical situation. We have to include that diversity, whatever that diversity might be. Maybe the patient moved in the middle of the procedure. We have to model that. And our future aspiration is that to get to this smarter level of realism. We are very realistic, but we want to be smarter. We want to have more diverse tasks and cases. We want to apply to broader set of applications. We want to go to other imaging modalities beyond CT, MR, cross-modality, and so on. Clinical applications, via digital twins, if I have digital twin version of the patient, I can apply them clinically as well. And eventually extending from VIT to what I call VCT. So not only predict the outcome of the imaging intervention, but actually outcome of the actual intervention all the way to the therapeutic outcome. For that, I need to have a biomedical models of the patients and models of the intervention. So I can say, you can manage this patient better, motion of the patient better, great. But you can also reduce the mortality by doing so by this amount. So I thank you for your attention. Thank you for inviting me to your meeting. I'm grateful to be with you. I really value your attention. Attention is the most expensive thing in the world today, I think. Thank you for giving me that honor. And I'll be happy to answer any questions you might have.
Well, thank you very much, Ehsan. We do have actually several questions. I think you've touched on many of these in your talk, so it may just be a further clarification on a few things. First question is, in what sense does a virtual virtual trial differ from simulation of experiments or from mathematical modeling of patients?
Not very much. You can say I'm putting repackaging the whole thing. I am claiming more from the simulation. Simulation has not, we have done simulation always. I have done, all of my career, I've done simulations. Because of that very reason that I mentioned, we cannot do experiment in medicine. So we have been doing simulations. Right. I am saying we can bundle our simulation with a high enough level of diversity and integrity to actually represent an actual clinical trial. Is a significantly higher claim for simulation. It's simulation on a steroid, if you will. That's what it is.
Next question was, assuming you had a successful virtual trial, what would be the next step after a successful virtual trial?
Well, I like to see that vendors rely on the virtual trial platform to condition their technology before actually bring it to the clinic, to design it right. So they would know not just this scanner is faster, therefore is better. Rather say using the scanner would enable to do detection of this thing or quantification of this or treatment of this 70% better. That's what I'm after. And the second thing is that in the process of doing so, if we can convince ourselves that we are doing it right, to make the process of approval of the technologies at the FDA level and commissioning much more straightforward. So we can put the technology in the best condition, the technology for the highest patient benefit. At the end of the day, we are here for the patient, not for the CT scan or the Linux that we got, right? So we need to make sure, in fact, I feel like this is our responsibility to make sure we best use that technology for that end.
A couple of other questions. One, are the resources like the XCAT model developed by Duke and the CBIT, are they open source?
We have not made them open source. It has been a massive philosophical discussion within our group. We love the concept of open source because it enables the community to engage. At the same time, sometimes I have seen things being open source and dropped. So it's like, what is the level of professionalism that you already need to have before you make it open source? And exactly how you condition it with the prospect. Open sourcing is not the goal. The goal is to make virtual trial a reality to improve medicine. Would open sourcing serve that function? If that is the case, we will do so.
Makes sense. One other question, can you simulate the vendor-specific recon algorithms, especially the DLIRs?
We are doing a fair amount of that already. The way we have done it is that we have our own reconstruction algorithm, but we cannot claim this is equivalent to Mr. X's reconstruction algorithm. So to do that, what we have done, essentially we have gotten a couple of reconstruction councils from the manufacturers, and with their aid and with their blessing, we hack them. So their reconstruction algorithms think our fake patients are actually real patients, and we run it through them. So we don't know what is inside the black box. We pretend we don't know it. And that way, though, the result that we get, we say is a reality of the actual what would happen in the clinic across the street. That's the way we have done it. It's somewhat limiting because you have to walk through the vendor. But I don't know what you can do if you don't walk through the vendor. I don't know any other way to do it.
Makes sense. I think we're at time. Anyone else have a burning question for Dr. Samei?
Actually, there is another question on the list. How you validate these very complex models?
Validation is an understatement for us. So there are validation at multiple levels. So because this is a very complex chain of things that you need to validate, for example, let's say I'm making a CT scanner. I need to validate the bow tie filter that I'm using. So the bow tie filter independently needs to be validated. So every piece at each stage is independently validated and verified. By verified, that means what? That means I think my bow tie filter shape is this way, but if I change it, would the result change in the way I'm anticipating? Does it make sense? If I increase the MA, would the noise go down? If the noise doesn't go down, there is something wrong with the way you have modeled it. So we essentially validate and verify at every step, and then we package multiple elements together. We say, okay, now I have a physical phantom, I'm going to image it. I'm going to also virtually image it. Now the resolution match. So if the resolution doesn't match, there might be zillion things under the hood that might be wrong. But I better have validated each component individually before I get to that point. And then one other thing I would say here is that not everything is going to be perfect, right? Because every time you do the experiment never matches the theory. We are physicists, we need to acknowledge that. So the question is that how much close is close enough? That's why I'm bringing back that concept of not only close to clinical reality, but also close for my validation. Because some things we don't want to waste your time to go from 2% accuracy to 1% accuracy. But if you go from 20 to 10%, that's a good way of using the stewardship of the resources that you have.
Thank you so much. Thank you again, Dr. Samei. This was really interesting. And certainly for an imaging physicist who knows next to nothing about this, I learned something new. So thank you.
Absolutely, it was great.
Therapy physicist, I should say. Sorry.
And we have multiple therapy participants, by the way, in the trial, as I mentioned. We are using it for all sorts of therapeutic applications as well.
Great. Well, thanks again. We will take the second half of that afternoon break, so about 9-10 minutes break.